Welcome back to the deep dive. You know, if you've ever tried to piece together a massive puzzle, but one where the pieces are these tiny, delicate fragments, you have a pretty good idea of the core problem facing spatial biology today.
That's a great analogy.
When researchers use spatial transcriptomics, or ST, to map out an organ, let's say a brain, they can't just analyze the whole thing at once.
Yeah, it's far too complex.
So the standard protocol is slicing the organ into these incredibly thin, parallel 2D sections. You can almost think of it like slicing a loaf of bread, right? Where each slice holds the gene expression data for its own little X and Y coordinates. But the critical problem is that the entire goal is to rebuild the original 3D loaf of bread. And in that slicing process, we lose the most crucial piece of information.
The z-axis, the exact order, and the height of each slice. You just have a stack of slices, and you don't know which one goes where.
Exactly. And that's not even the only problem.
Not by a long shot. This loss is compounded by technical difficulties. You're dealing with molecular data, and every single one of those slices is processed and sequenced independently.
Right, in its own little batch.
Which creates these systematic technical variations, what we call batch effects, in the gene expression profiles. So even if two adjacent slices were biologically identical, they end up looking slightly different just because of the lab work.
So you end up with a pile of beautiful, high-resolution, but totally flat maps that are noisy and don't know where they belong vertically.
A beautiful, noisy, disorganized pile of naps. That's it.
In previous computational solutions, there were really just partial fixes, weren't they? Some, like precast or paste, were great at 2D alignment, but only within a single batch.
And others that tried to do 3D, something like Stitch 3D, they had a major catch. They required you to already know the z-coordinates.
Which kind of defeats the purpose if you're trying to build a new reference atlas where those coordinates are missing, especially if you're pulling samples from multiple different patients.
Precisely. The field was waiting for a truly unified end-to-end solution, something that could handle the molecular integration and the spatial alignment. But the real key, you had to figure out that missing z-coordinate completely unsupervised.
Relying only on the data itself.
Just the data.
Which brings us to the focus of our deep dive today, STAIR, spatial transcriptomic alignment, integration, and 3D reconstruction. So today, we're going to unpack STAIR's two powerful core modules, STAIR-M for the molecular side and STAIR-LOC for the physical coordinates, and look at the really compelling evidence from benchmarks covering the human brain, the mouse brain, and even clinical breast cancer samples.
Fantastic story.
Okay, so let's start at the beginning with STAIR-M, the integration engine. Before we can even think about 3D, we have to make sure the molecular data across all those slices is speaking the same language. That means tackling the batch effects. Why is dealing with this noise the absolute first priority?
Because if you don't clean the data first, any spatial learning that comes after is just building on a foundation of sand. It's garbage in, garbage out.
So the noise just gets amplified.
Exactly. When spots share information with their neighbors in the graph, they just propagate that batch noise. So STAIR uses what's called a batch-aware autoencoder to handle this. You can think of an autoencoder as a highly efficient filter.
Okay.
It takes your really high-dimensional gene expression data and it compresses it into a cleaner, lower-dimensional feature set. But it's specifically designed to identify and filter out that technical variation, the noise that was introduced by the batch.
So it's not just compressing the data, it's actively learning what the real biological patterns look like and separating that from the noise.
That's it, precisely. It uses a very specialized statistical approach that's tailored to clean up the messy, sparse, and often zero-heavy data you get from transcriptomics. This whole process harmonizes the features before any spatial learning is even attempted.
So once those features are clean, the system gets organized using a heterogeneous graph attention network, or a GATE. Graph networks are great for defining relationships. Walk us through how this huge multi-slice graph is built.
Okay, so you treat every single data point, every spot, across every single slice as a node in one gigantic network.
Every single one.
Every one. The crucial part is defining the connections, or the edges. First, you have the intra-slice edges. These are simple. Spots that are physically close together within the same slice get connected. It's just a standard nearest neighbor rule.
Okay, that makes sense. They're physical neighbors. But how do you connect a spot in, say, slice 5 to a spot in slice 15 when you don't actually know their physical distance yet? Those are the interslice edges.
Right, and those are connected purely based on their molecular identity. If two spots from different slices have highly consistent corrected gene expression features, if they are molecularly almost identical, The system draws an edge between them.
I see. So it's using the biology to bridge the physical gap.
Exactly. This lets the graph implicitly learn the semantic or functional correspondence across the entire stack, even when the slices are far apart.
Which brings us to what feels like the core innovation of STAIR, dual-level attention. The system has to figure out which of these thousands of connections are actually important. What are these two levels?
So the first level is pretty straightforward, spot-level attention. This just tells you the importance of one individual spot to another individual spot, either within its slice or across slices.
Okay.
But the real breakthrough is the slice-level attention score. This score measures how much the entirety of one slice contributes to the structural definition of another slice. It's capturing these higher-order semantic relationships.
So instead of just saying, this spot looks like that spot, the system can say slice A as a whole is functionally very similar to slice B, and that structural continuity is really important.
That's it. It's capturing the global organizational patterns. And that slice-level score, it becomes the absolute linchpin for everything that follows, as we're about to see.
So let's look at the evidence. The 10x Visium dataset from the human dorsolateral prefrontal cortex, the DLPFC. They used 12 slices across 3 separate brain samples, right? Testing STAIR's ability to correctly assign spots to the known anatomical layers.
And the results were, well, they were definitive. When integrating all 12 slices at once, STAIR achieved an integration accuracy, which we measure with something called the adjusted RAND index of 0.65.
And how does that compare?
That's a massive improvement. Older methods like STAIR, they peaked at around 0.46. It just means STAIR was far, far more successful at grouping molecular spots into the anatomically correct layers, even with all the batch differences.
But here's the part that really got me. It wasn't just confirming the known biology. It was accurate enough to actively refine the existing annotations.
That was the real aha moment. In one of the samples, some spots had actually been misclassified by pathologists during the original manual annotation. STAIR identified this small, distinctor group of spots that should belong to layer 1 and layer 2.
Which had been mistakenly grouped into layer 3.
Exactly. And the researchers then used known layer-specific marker genes, like GFE, for layer 1, to biologically validate that STAIR's computational refinement was actually correct.
So it was using the molecular features to correct human error.
Huh.
That's incredible.
It is. The overall takeaway from STAIR is just this really strong integration capability. It led the field in the ASW F1 score, which is a metric that basically judges how well you separate the true biological zones while minimizing that technical batch variation.
Right, so STAIR has successfully cleaned and aligned all the molecular features. But that just gives us a collection of aligned molecular maps. We still need to solve the fundamental problem, figuring out that missing z-axis position. If earlier methods needed that coordinate as an input, how does STAIR infer it completely unsupervised?
This is where that crucial slice-level attention score we just talked about becomes the structural blueprint. Remember, that score quantifies the semantic relationship, how much the entire molecular makeup of slice I contributes to the definition of slice J. STAIR makes a very simple but powerful assumption. High molecular similarity means close physical proximity.
So the molecular coherence is a proxy for physical closeness.
Exactly. They convert those attention scores into a semantic distance metric. If 2 slices have high mutual attention, their semantic distance is low. They're close.
And then what do they do with those distances?
They use them to build a structure called a minimum spanning tree, or MST, using an algorithm called Kruskal's.
For anyone not familiar with that, how should we visualize a minimum spanning tree here?
Think of it like a roadmap. You have all your slices, those are your cities or your nodes, and the semantic distances are all the possible road lengths connecting them.
Okay.
The MST finds the shortest, most efficient way to connect all the cities without creating any closed loops or circles. By defining this single optimal path of connection, the MST implicitly establishes the sequential order and the relative vertical separation of all the slices. And just like that, you have your inferred z-axis coordinates.
That is just. It's ingenious. Using the internal molecular relationships to create external spatial coordinates. How did they validate this actually works?
They use a perfect test case. 12 merfish slices from the mouse hypothalamic preoptic area. And crucially, this data set had the true measured z-coordinates. So they had the answer key.
And how did it do?
Testing STAIR's attention-based reconstruction against that ground truth. It yielded virtually perfect results.
Perfect? What do you mean by perfect?
I mean, the Pearson correlation coefficient, which measures how accurately the positioning matched the known order, was 1.0.
A perfect correlation.
The normalized mean squared error and the misordering rate were both 0.
Zero error. So it got the order exactly right and the spacing perfectly correlated.
Exactly right. Near perfect positioning based purely on molecular attention.
And the method proved to be incredibly tough. They tested its robustness by randomly cropping up to 60% of the data points from the slices. I mean, losing over half your data is a massive hit.
It's huge. And simpler metrics, like just correlating overall gene expression, they degraded very quickly. But the attention-based method remained highly robust. Even with 60% of the spots gone, the error rate was still negligible.
Which confirms that the dual-level attention network isn't just relying on local details.
That's right. It captures these really strong, higher-order semantic associations that persist even when the data gets extremely sparse.
And to see this scaled up, they successfully applied STAIR to reconstruct a whole mouse brain atlas. 40 coronal half-brain slices along the entire anterior-posterior axis.
And the 3D reconstruction was highly accurate. They validated the whole thing by checking for biological plausibility. They looked for region-specific marker genes. For example, they confirmed that genes like DSP mapped correctly to the hippocampus, GPR88 mapped to the striatum.
All exactly where the anatomical map would predict in the new 3D volume.
Exactly.
So STAIR has proven it can take molecular features, clean them, integrate them, and then use those relationships to perfectly order and position slices in 3D, entirely without guidance.
That's the core of it, yes.
Now let's turn to STAIRlock, the module responsible for that precise 2D, so the XY alignment. If we're already integrated, why do we need a whole dedicated module just for 2D registration?
Because even if your slices are perfectly ordered, they might be slightly rotated or translated relative to one another in that XY plane, just from how they were placed on the slide.
A slight shift.
A slight shift. So STAIRlock handles this with a two-step refinement process. The initial alignment uses those cohesive spatial features from STAIR-M to find what are called mutual nearest neighbors, or MNNs. These pairs give the system a good first guess for the rotation and translation needed, a strong starting point.
And then comes a fine-tuning.
Exactly. The final alignment uses the well-known iterative closest point, or ICP algorithm, but it applies it intelligently. Instead of just throwing every single data point at it, STAIR focuses only on the informative spots.
Which are what in this case?
Primarily the concave hulls. You can think of those as the molecular boundaries of the defined tissue regions and the overall slice boundaries themselves.
Why are the boundaries so important for alignment?
Well, geometrically, if you align the boundaries of two objects perfectly, the interior is going to align naturally. If you only try to align scattered spots in the middle, you risk warping the whole structure. By preserving the shape of those molecular compartments and tissue contours, STAIR ensures the registration is both precise and biologically meaningful.
And the results on those shuffled Murphish slices were fantastic. Median translation errors of just 0.04 millimeters. I mean, that is incredibly precise for this kind of data.
It is. It's proof that combining strong feature learning from STAIR-M with smart geometric registration, so STAIRlock focusing on boundaries, gives you superior 2D accuracy.
What's also fascinating here is STAIR's ability to bridge technologies. They tested cross-platform integration using mouse olfactory bulb data from two very different sources, StereoSec and SlideSeq-V2.
Right, and those are vastly different. StereoSec offers sub-single-cell resolution, while SlideSeq-V2 is near cellular. We're talking different data scales, different gene counts, a whole different ballgame.
But STAIR handled it.
It did it. It successfully integrated them. And crucially, the framework was able to identify the shared anatomical regions, like the main olfactory bulb, and align them coherently. But it also correctly preserved the unique biology that was only captured by one platform. For example, certain subregions that were only sampled by the SlideSeq-V2 data.
So it integrates what's common, but respects what's unique.
Exactly, which is vital for building robust reference atlases from many different sources.
Let's move to the clinical relevance. Specifically, the HAIR2 positive breast cancer case study. This involved 3 consecutive slices. What new critical biological knowledge came out of seeing this in 3D?
This is where the 3D vision truly shines. STAIR identified 8 distinct spatial regions within the tumor, distinguishing areas that other methods had just lumped together. Most importantly, it clearly differentiated 2 distinct types of in situ cancer cancer 1 and in situ cancer 2, and also mapped out a highly heterogeneous fibrous tissue right next to the tumor.
And with these defined 3D boundaries, they could actually start to trace the disease's progression.
Absolutely. By using trajectory inference methods, they established a molecular progression path. In situ cancer, too, showed lower malignancy and high immune activity. That then progressed into in situ cancer 1, which showed stronger malignancy, high in key cancer drivers like ERBB2, which then led, eventually, to invasive cancer.
And the 3D reconstruction provided the final piece, heterogeneity along the z-axis. The tumor invasion wasn't just a flat process.
That's right. The 3D view revealed that the most aggressive regions, the invasive cancer, and that in situ cancer 1 showed a distinct trend of progression from slice H3 up towards slice H1. This interslice heterogeneity is the crucial context we miss in a 2D slide. It shows how the disease is spatially invading the tissue in 3 dimensions.
Okay, so we've established the accuracy and the utility, but STAIR is also designed for atlas expansion. If a researcher generates a new slice, we don't want to have to rebuild the entire 3D atlas from scratch every single time.
Yeah.
How does STAIR do this?
It leans heavily on that structure that's already defined by the slice-level attention. So, let's say we have our existing 40 slice mouse brain atlas, and we introduce a new Visium slice.
Right.
STAIR integrates the new slice, and its z-axis position is predicted using an attention-weighted average based on all the existing slices in the atlas.
So if the new slice is most similar to, say, existing slices 19 and 21, the attention scores will just automatically place it right between them.
Correct. And once the z-axis is predicted, the nearest atlas slice, let's say slice 20AX, is the 2D template. STAIRlock then aligns the new slice's X and Y coordinates to that template, and that instantly transfers all the existing structural annotations.
So things like the common coordinate framework coordinates and Allen Brain Atlas regions just get mapped right onto the new data.
Instantly. It drastically simplifies the process of creating these massive collaborative atlases.
But every robust computational method has its limitations. This is where we need to apply some critical thinking. For all its innovation, what are the constraints researchers should keep in mind with STAIR?
There are a few key ones. First, STAIRlock relies on the assumption that the global tissue contours are preserved across slices. If that continuity is broken, say, there's significant manual dissection or very sparse overlap between sections, the alignment accuracy will drop.
And there's the geometric assumption. The model treats the tissue sections as being perfectly parallel.
Yes. That parallel assumption really simplifies the math for the unsupervised z-axis inference. But, real-world tissue prep often results in non-parallel sections or slight unknown cutting angles. Future versions will need to handle that geometric variability.
And finally, there's the problem that plagues all of these kinds of reconstruction methods. The banana slicing problem.
It's a great visual, isn't it? When you reconstruct a naturally curved structure like a brain or a developing organ from flat cross-sections, the current methods tend to straighten the overall structure out.
So you get a straightened banana?
Exactly. The reconstructed 3D atlas has this slight linearization. To fully recover that natural anatomical curve, STAIR would need some kind of external geometric prior, like MRI data, which it currently doesn't use.
And we also noted the scalability issue.
We did. To accurately calculate those global slice-to-slice relationships, those slice-level attention scores, you currently need to retrain the model jointly with the existing atlas every time you add a new slice. And that can get computationally expensive for truly massive data sets.
Okay, so what does this all mean for us? STAIR really represents a pioneering and unified approach. By using a batch-aware embedding and this dual-level attention in a heterogeneous graph, it's the first framework to successfully build accurate 3D molecular atlases from serial 2D slices alone, without any prior knowledge of the z-axis.
And by providing this critical 3D molecular context, STAIR doesn't just rearrange data. It fundamentally changes how we can comprehend complex tissue organization. It lets researchers move far beyond the limits of a flat 2D slide to explore molecular mechanisms, disease invasion, and development in their native structural environment.
We saw how those slice-level attention scores, derived from the dual-level attention network, were so effective at predicting z-axis positions and even proved robust when data was sparse. So when you're considering highly curved structures like the gut tube of an embryo and tackling that banana slicing problem, which of the two primary STAIR components, the batch-aware nonlinear embedding or the dual-level attention network, do you think would be most crucial in helping a future version of STAIR finally recover that natural anatomical curvature? That's your deep dive for the week. We'll see you next time.